Okay. So today we are going to talk about loudness. We talked about pitch. And on Wednesday we'll talk about sound localization. Let's see if we finished on Wednesday or we continue on the last day of the semester on next Monday. Hi, Charles. So pitch and loudness are psychological phenomena. They are internal interpretations of sound frequency in the case of pitch and sound wave amplitude in the case of loudness. And as we have been seeing again and again, they are only indirect interpretations that are only related to the physical world. And in many cases, they are not more than weakly, flimsily related to the physical stimuli. So we are going to talk about two complementary hypotheses of how our system codes amplitude. And then we are going to talk about critical bands, which is important. It's not in the book, I don't know why. And it's very important to understand loudness. And it's a little bit complex, but not complicated. So when we talked about sound, can you see my mouse when I hover? Yes, okay. So when we talked about sound, we saw that the range of amplitudes of air pressure waves that we humans perceive is enormous, like a jet, an airplane taking off emits a sound that is 10 million times more intensive than the quietest sound we can hear, the minimum threshold. And that's why we prefer to represent amplitudes of sound waves on a logarithm scale with units of decibels. And that allows us to have a more intuitive grasp of differences between sounds that are common in our daily life. So on a linear scale, the difference between a quiet noise, let's say leaves rustling, compared to a loud noise like a subway train, which is 10,000 times larger, is uncomfortable. On a log scale, on decibels, this comparison is more manageable. So it's not 10,000 times larger, it is 80 times, it's between 20 and 100. Even normal conversations, our voice, the amplitude of our voice fluctuates up to 10 times. And this huge range of amplitudes is an important factor that we have to account for in order to understand how our system analyzes and encodes differences in loudness. So here we already have the first evidence suggesting that our perception of loudness is only related, but it's not perfectly, far from perfectly correlated with sound waves intensity. And we can see it in the fact that these lines are not straight. So all these sounds here, across all frequencies, they have the same intensity, 60 decibels in this case. But look what happens in our perception. Depending on the frequency, loudness is perceived drastically different. So we'll see two hypotheses, two propositions that try to explain this relationship. But first, let me remind you of some things that we already saw, that we need to refresh in order to understand loudness. So this is the ear, our auditory canal, and then this is the eardrum. Now here we have the inner ear with the three ossicles. This is the stip with its foot plate transmitting the air vibrations into the cochlea, into the liquid, the fluid inside the cochlea. So this is the cochlea unfolded, and across the cochlea in the middle we have the basilar membrane. That these are pure tones, right? And in natural sounds, complex tones shut up. Thank you. So in complex sounds that are all of our daily normal sounds, they comprise many different frequencies inside. So any sound that reaches our ear will oscillate the basilar membrane at many different places and at the same time. So last time when we talked about pitch, we covered two complementary hypotheses. The place theory suggests that our system interprets pitch according to which area of the basilar membrane oscillates. And the temporal coding theory suggests that irrespective of which hair cells, which area of the basilar membrane oscillates, our system discerns pitches according to the behavior, the firing rate of the hair cells that are firing. They are not, these two theories are not mutually exclusive. They work at the same time, right? So according to the different frequencies comprising any given complex sound, specific areas of the basilar membrane oscillate at the same time. And in these areas, and only in the areas that oscillate according to the sound, the tectorial membrane, so this is the basilar membrane and it oscillates, and we have the hair cells longitudinally across the membrane. This is a cross section, yes. So wherever the basilar membrane oscillates high enough, the tectorial membrane rubs the tipplings, the cilia that are on the top of the hair cells. And then when it rubs the cilia on the top, there are those tipplings that connect between the cilia, and then just like a light switch, every time the tipplings bend, the hair cell fires. And we saw that it works exactly like a light switch, on off, meaning firing, not firing, that is synchronized with the sound frequency. So this is the sound, so it fires, not fires, fires, not fires. Now, what is synchronized is the on and off, the firing, not firing. not the firing rate itself during the interval that each cell fires, okay? Which means that these cells can fire according in synchronization, in locked phase with the sound, but it can fire with a lower firing rate, which would mean less spikes during this interval, or a higher firing rate, more spikes here, okay? So the firing rate hypothesis suggests that the perceived loudness of a given sound is a function of the firing rate of the neurons that are firing when they fire. So what we have here are a pure tone, the same frequency, so they are perceived in terms of pitch, they are perceived identically, but they have different amplitudes. Here we have the auditory nerve activity, so they fire in the same frequency as the sound, but for a higher amplitude they fire faster during the interval in which they are firing. All right? Is it clear? It's a nuance, not complicated, but it sometimes confuses. Okay. But we still have a problem. We still have a question to solve. Our neurons, they are limited in their dynamic range of firing rates. They cannot, and we know that that the range of amplitudes of sounds that we hear is endless, is huge, and each neuron does not have such a wide range of firing rates. So the answer is that hair cells, they not only specialize in specific frequencies, but they also specialize in specific ranges of sound amplitude. So OHC is outer hair cells and IHC is inner hair cells. What we see here is this additional subspecialization of these inner hair cells that are tuned to the same frequency. They are neighbors, they are in the same area on the basilar membrane, but they are sensitive to different ranges of sound intensity. So here, this guy will change its firing pattern when its preferred sound frequency ranges between 20 decibels. This is neuron response, yes, and this is amplitude. These are all for the same sound frequency. So this guy will change its behavior when the sound is between 20 decibels and 55 decibels. And this guy, for the same sound frequency, will only change its behavior when the sound is for intensities between 50 decibels and 80 decibels. So even though each cell by itself cannot cover the entire range of intensities of sounds, if we combine the activity of different hair cells for the same frequency, if we combine them together, we have a range of different firing patterns that cover the entire range of amplitudes. In this case, these two hair cells together, they cover between 20 decibels to 80 decibels. All right? And it's the same, it's similar to the HDR function that we all have, that you probably all have in your smartphone camera, that the camera, all cameras, when we had the film, we had different, never mind. So any camera has a maximum dynamic range of light. So what the HDR camera does, it takes different pictures with different exposures, meaning that a low exposure, little light will enter through the shutter, and the lightest areas will fall within its best dynamic range. So we'll have a high resolution within the lightest, the parts with most light, but all the darker part, less light, will be completely dark. And then we have middle range exposures, and the largest exposure, a lot of light enters, meaning that it will burn all the parts with a lot of light, but the darkest parts will fall within its best dynamic range. So we can see all the details here. So what the camera does, it takes for each one of those different pictures with different exposures, take the part of the picture, with the best dynamic, with the best resolution, combine them together in one picture. Okay. All right, the second hypothesis is the number of neurons hypothesis that complements the firing rate hypothesis. The idea is very simple, that the louder the sound, the more neurons, the more hair cells will fire at the same time. Okay? So here again we have the same frequency. This is louder. This is a higher amplitude. So not only this auditory nerve will fire, these guys will start firing as well. Okay, we already saw this. So here we have the frequency tuning, the preference frequency for single hair cells recorded from a cat. Here we have the frequency, and here we have the threshold, the minimum sound intensity in decibels for that specific hair cell to start firing. And we can see that all of them have a very narrow range of sensitivity for lower intensities, and the higher the intensities, the wider it gets. Okay? So let's take this cell. Any point that falls within this area, this hair cell will fire, right? For this cell, any point in this area, you can all see it, right? So let's take a 4000 hertz tone. We can see that up to, let's say, 58 decibels, it will fall beyond the area of only this cell. So only this cell will fire for 4000 hertz up to 58 decibels. at 4,000 hertz at 65 decibels, not only this cell will fire, but suddenly also this cell will fire. And if it rises to 80 decibels, all these 4 cells will fire. Now let's take 800 hertz up to 35 decibels. Only this guy will fire, but a pure tone of 800 hertz at 80 decibels, all these 7 cells will fire. So the louder a sound with any given frequency, the more neighboring cells will start firing. Now I forgot to mention, these cells, they are because this is frequency, and we know that the basilar membrane is organized by frequency, spatially organized by frequency. The closer they are, the closer the frequency. So they are. This is also the same spatial relationship between them. So the louder, the more intense, the higher the amplitude of any given sound, the more neighboring cells will start firing. And that explains why when we move from low intensity sounds, the louder the sound, the straighter the line. Okay, because more cells are firing. Is it clear? Interrupt me anytime. Okay, now this evidence was what I just shown you. These are single cell recordings from a cat. So the question is, how can we connect electrophysiological evidence to human perception? We cannot do single cell recordings like this in humans. So we have to resort to psychophysics to see if the same patterns can be connected to any behavior, observed behavior in humans as well. I remind you, so this is the Fourier representation. So this is a pure sound. This is a violin tone that comprises many different harmonics. And this is white noise, meaning a white noise comprises all frequencies, all at the same amplitude. Yeah, sorry, I forgot to tell you. Excuse me, I forgot to warn you. Please be prepared to adjust your volume. So this is a pure tone, yes, sorry. And this is a white noise. No, this is not a white noise. Okay. You remember how a white noise sounds, like it has no pitch. Now, what we have here are different ranges of white noise. What time is it? Okay, we have time. Different ranges of white noise. That is, they comprise all the frequencies under this graph, and all of them have the same frequency. So the narrower the band of a white noise, the closer it will sound, it will be perceived as a one pitch. So if it gets narrow enough, it will be like a pure tone. Yeah, makes sense? Okay, let's see how it sounds. So here it will be from top to bottom. Okay, this is the same as this one. I have a mess here. So what we just heard is this. Okay, I'll do it again. Now, apparently my thing is here. So now we'll hear this, and this. Sorry. So this was this. Okay. Now we'll hear this, and this. No. Now we heard this, and this. Okay. Can you understand what we're hearing? Okay. The last one we heard is the same width, but centered on different frequencies. Let's try, and this, one of them is wider frequency, narrower, and very narrow. Let's see what happens if I touch here. This was this one. This was this one. Okay? It's clear, right? No! Okay. Now, we already saw that in vision, there is a summation and integration of energy when it's presented within a very short period of time. So all this is vision. Yes, we have time here, and we have luminance, the amount of photons that reach our retina. So all these 6 graphs, they have the same area under the function. So they have the same physical energy. And when any one of these are presented within a window of 100 milliseconds, they are perceived identically. We cannot discern between them. So the question is whether our auditory system also works like that. So what we have here are sounds that have the same physical energy. They all have the same area under the function. All of these are referenced, they are identical. And here below, we have different distributions of the same area in different ways. Concentrated in a narrow range of frequency with high amplitude, and then it widens and the amplitude goes lower. So they all have, so energy, physically, energy is the square of the amplitude of any wave. So when, this is just a note, so when you triple the amplitude of a wave, the energy increases by 9 times, which is 3 squared, yes? But here it doesn't matter because they all have the same area, they all have the same energy. And they are all centered on the same frequency, okay? They are all around the same average frequency. So the question is whether we perceive differences in loudness between these guys. These are identical. So in other words, the question is, does our perception of loudness is also based on summation and integration of the physical energy of a sound stimulus? So if yes, all of these should sound equally loud as this, okay? So what we are going to hear now, and I ask you to participate, write down, you will hear 8 pairs of sound. So every time it will be the reference, which is the same for all the 8 pairs. You hear the reference, and then the test sound, and then again the reference and the test sound, the reference and the test sound. We hear every time the reference, as the word says, to have a reference every time. Because if we hear every time each one of them, it's hard to compare between all of them. So we every time compare to the same reference. So you hear here there are only 5 pairs, but what you hear are 8 pairs. So write down for each pair whether you hear the test, the second sound, as quieter, the same loudness, or louder than the reference sound. Okay, are you ready? Write down from 1 to 8. Quieter, same, or louder.
Critical bands by loudness comparison. 8 times you will hear a reference noise band followed by a test band of increasing width and identical power. Compare the loudness of reference and test bands. The demonstration is repeated once.
Okay? Who wants to write in the chat or say aloud his or her results? No, okay. I framed the question not well. I know nobody wants. Who is ready to do me a favor and share with us the results? Okay.
Yeah, like the last 3 ones are louder to me.
Yes. 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 Perfect. Everybody got... You are all functional auditory systems. Thank you. So these are the usual results. Now this is a schematic exaggeration, okay? This is only for us to consider it conceptually. The function is supposed to be more gradual and more, smooth. So what should we see here if we had perfect integration and summation? We should see a straight line, right? The same loudness across all the 8, we have here only 5, across the 8 pairs of stimuli. But as you guys perceived, the first ones sounded the same loudness, but suddenly it started to increase. Why this increase and why sudden? Okay. So let's begin with the increase, which is easier to understand. So we saw that the basilar membrane, the more frequencies a sound comprises, the larger the area of the membrane that oscillates at the same time. And the more frequencies a given sound comprises, the more hair cells will fire at the same time. And as we saw, the number of neurons hypothesis suggests, and this is evidence supporting it, that the more hair cells firing, the louder our system, our brain will interpret the sound, even though it's the same physical energy. So far, so good, right? But why this flat area? And now we have to understand the idea of critical bands. So, these are pure tones, okay? Now, this is only one, as much as technically possible, this is only one isolated frequency. But because the basilar membrane is one continuous piece of tissue, there is a limit how much any part of the membrane can oscillate in isolation without at the same time affecting at least its nearest neighboring areas, right? It's one piece of thing. It cannot, you understand. So let's say the oscillation, the oscillation of a specific area, let's say that responds to a 2500 hertz pure tone, will end up affecting the areas, the neighboring areas that respond to 2450 hertz, and also the area that responds to 2550 hertz. So the narrowest, the smallest area that a pure tone ends up affecting is the critical band. Okay? So a sound, a pure tone sound of 2500 hertz with a given amplitude will affect the same area as a sound that contains frequencies ranging from, so this, let's say, is 2500, a sound with the same energy that it's spread on a larger area, if it remains within the critical band, will oscillate the same area of the membrane. We will remain oscillating the same area of the membrane. Now, if we have a pure tone, the higher the amplitude, the larger the area it will oscillate, right? So a minimum, a pure tone, very small, will oscillate the minimum critical band, and then a higher amplitude will remain only that critical band. At a certain point, it will start oscillating more and more neighbors. Okay, so as long as the amplitude, as long as the critical band is not expanded, if we have a lot of, and here we can see, if we have a lot of small amplitude frequencies, neighboring frequencies with small amplitude, they will oscillate the same area of the basilar membrane as a narrower range of amplitudes with higher frequency, with higher amplitude. Is it clear? I confused my words, but is it clear? Anyone? Make sense? Even though these two will sound a little bit different in terms of pitch, in terms of timbre, but in terms of loudness, they will sound the same. So the elbow here represents the critical band for this specific frequency. This specific frequency is the average, is the center of all these ranges. Okay, so here we have from here, these guys, this additional frequency started to oscillate additional larger and larger areas of the basilar membrane, even though it's the same physical energy, and then our system started to interpret as louder and louder. Okay? All right. So here we have the basilar membrane. Heliocotroma is here it's the apex. Okay, here is the base, and here is the apex. In the base, we have hair cells that react more, the largest oscillation of the membrane when we have high frequencies, and here low frequencies. So what we can see here is that along the entire basilar membrane, the critical band, physically, the critical band for any pure tone is the same, is 1.3 millimeters. So the quietest sound, the quietest amplitude for any pure tone that we can hear that is above our minimum threshold will oscillate at least 1.3 millimeters of the membrane. And this is the critical band in spatial terms. In frequency, in sound frequency terms, the critical band for any given pure tone, for any given frequency, is something around between 10 to 15% of that pure tone. So for 1,000 hertz, the critical band is 100 hertz, meaning that a pure tone with 1,000 hertz, with a given energy, will sound the same in terms of loudness, will sound as loud as a sound with the same energy that is spread from 900 to 1,100 hertz. Okay? For 8,000 hertz, the critical band is 1,000 hertz. So if we maintain the total energy, so an 8,000 hertz pure tone with a given amplitude will sound the same as a combination of lesser amplitude frequencies between 7,000 and 9,000 hertz, as long as they have the same physical energy. Okay? Now, the louder okay, now that I said already, I will not repeat myself. As the energy of a noise stimulus is spread across a wide range of frequencies, the sound, yes, anyone? It's easy, no? Talk loud, yeah? B grows louder, but this is not an accurate wording because it will tend to grow louder, because it will only be perceived louder beyond a certain threshold. If it starts to oscillate an area larger than 1.3 millimeters, wider than the critical band of that noise. Right? You all agree with me. Where are we? Here. Okay. Now this is a second. Now what we saw before was the idea of the interpretation of critical bands applied to a discrimination task, and this is a matching task. Okay. Okay. Now I'll say it right instead of wrong. So the previous one was a matching task. This is a discrimination task. So what we have here, these are called the pedestals. Okay, so we have a white noise, all the same amplitude with wider and wider ranges of frequencies. So from top to bottom, they will sound, all of them will sound louder. I don't know if from here to here we already, if we are already beyond the critical, the critical band. Maybe this and this will sound the same. But if this is beyond the critical band, then yes, they will sound louder and louder. And then, to present it together with the pedestal, we have a pure tone. All of them centered in the same frequency. This is a pure tone. And the question is whether we can discriminate the pure tone that is presented together with the pedestal, whether we can distinguish between these two sounds, okay? Now, what you hear is not the experiment. The experiment is for each of those, you hear many different intensities of the pure tone, of the test tone, until the participant says, yes, I hear the difference, okay? But this is only an example, so for you to see how it sounds. Okay, it was the pedestal and the pedestal plus the pure tone. So see if you can distinguish between the difference between these two, between pedestal without and pedestal with. The third one is the pedestal is getting wider and wider. Okay, well. So this is again, it's a schematic exaggeration. So in the previous one, the matching one, we had all the stimuli had the same energy, and here we have larger and larger energy, more and more energy. So and they all have the same amplitude. So from left to right, they also sound louder and louder, and you heard it, hopefully. And what we see, and the function is the minimum intensity that the pure tone must be sounded, presented together, in order for the participant to discriminate between pedestal without the pure tone and pedestal with the pure tone. Okay? So, for narrow pedestals, the wider the pedestal, the louder the pure tone had to be presented in order to be discriminated. But suddenly, beyond a certain width of the pedestal, even when the pedestals got wider and therefore louder, the pure tone was discriminated without it needing to be presented louder. So this sounded louder than this, but the pure tone could be presented at the same intensity, and it was equally discriminated. How is it? So let's begin here. So the first three pedestals, they remained within the critical band for that frequency, which means that they affected the same area of the basilar membrane. So they sounded similar in terms of pitch. So they masked the pure tone similarly. So the louder they got, the louder, the more intense the pure tone had to be presented in order for the participants to discriminate. Okay, so far so good? Yes, makes sense? Good. But beyond a certain point, which is beyond the critical band, the addition of more frequencies not only caused the noise to sound louder, but it became more and more different in terms of pitches and timbre, different enough for the pure tone to be discriminated irrespective of how loud the pedestal sounds. So beyond a certain point, the amplitude, the addition of frequencies did not affect the discrimination of the pure tone. It would be like if you have, let's say, if you have an adult, a male adult with a bass voice, talking among other people, other adults with also bass voices, it will be harder to discriminate that voice. But if this adult is talking together with children, the same intensity, even children shouting louder than the other adults, that adult will be it will be easier to discriminate the adult's voice. Okay, I don't know if this example is exactly accurate, but you got the idea. It helped. Okay, I'm happy that it helped. So the concept of critical band is related. It's not the same, it's not the cause, it's not the consequence, it's only related to what we see here, the width of the range of the minimum threshold for any given cell. Like any given cell, for low intensity sounds, will react to a certain range, to a certain band of frequencies. This would be commensurate to the critical, meaning that, let's say that when this hair cell was born, God told this hair cell, you are 5,000 hertz only. Yeah, but what happens is that when 4,000 hertz is presented, it's the neighbor, but the neighbor also ends up oscillating this hair cell. So for quiet sounds, let's say between 3000 and 6000 hertz, only this cell will no, the opposite. For quiet noise sounds, this cell will be activated only by this narrow range of frequencies. But above a certain intensity, all the basilar membrane, let's say, look here at 200 hertz sound, if it's loud enough, the entire basilar membrane will oscillate and it will activate even this far away hair cell. Okay? So this is the basilar membrane again. We have here stretched, these are the hair cells, and this is the auditory nerve. The base with the oval window and the apex here. So we have A, B, and C are 4 different sounds that when they are presented in separate, they are easily discriminated one from the other. Let's say they are pure tones. Is it written? Well, we can assume these are pure tones. And when they are presented in separate, they are easily discriminated one from the other, and they are also perceived with the same loudness. But if A and B are presented together, they will have a considerable shared area that both will oscillate. So it will be difficult to discern between A and B when they are presented together. When C and D are presented together, they are easily discriminated because they affect different areas. All these 4 sounds, they have the same, they are perceived as the same loudness. It doesn't mean they have the same amplitude, because we saw already that different frequencies need different amplitudes in order to be perceived the same loudness. Okay, so this is not the same energy, it's the same loudness. So we can assume by looking here that this had much more energy than this, and the 3 of them, because for a very low frequency sound, in order to be perceived as the same loudness as sounds here and here, they have to be with a much higher amplitude. You remember the graph, right? That goes like this. So these, when they are presented together, they are hardly discriminable, and these 2 are easily discriminable. At the same time, for the same reason, when A and B are presented together, they will sound quieter than C and D presented together, because the added overall area affected by C and D is larger than the overall area affected by A and B together. Good? Okay, so this is a written summary of critical bands. There are 2 points that I want to stress here. Loudness depends not only on the amount of energy in the physical stimulus, but also the number of critical bands activated, affected by that sound. That is, not only the amount of energy, but also the number of cells firing at the same time. And secondly, 2 sounds with equal energy but different bandwidths only have equal loudness when they are both confined to a single critical band. And that's because if they are confined within within a single critical band, the same number of cells will fire at the same time in both cases. All right?